Okay, let's start. Good morning, afternoon, evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us the 73rd seminar, and I call it a sprint day. A recent article in Nature made me think and discuss with my colleagues. This paper argues that papers and patents are becoming less disruptive over time, and this is unfortunately true, partly due to the current academic culture that emphasizes near-term returns on investment. Some academic leadership people keep counting grant amount and the number of papers, not the real impact of faculty research and education. If you ask the return on investment, although I hate to hear the term, in a non-profit educational organization, the return should be well-trained future leaders and innovation. Luckily, our future generations inspire me with creative ideas and activities, so I am very optimistic. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Dr. Patrick Rose. I got to know him as an ONR Global Program Manager. I have an ONR project supported by Sandra, my current program manager. She and I share the same crazy vision, and Patrick and his awardee Mark have been discussing a collaborative project that may have a huge impact if possible or successful. Patrick recently moved to SPRIND, the German Federal Agency for Disruption, and I believe that he cares about innovation and disruptive technology. Patrick, I truly thank you for your time and for your contribution to the scientific community, especially by supporting innovative ideas and projects, and the virtual podium is all yours now. Thank you.
Thank you, Taesuk. It's an honor to be part of this program, this movement that you've created. I think it truly is a movement, and congratulate you on what you've been able to accomplish and achieve. And I have no doubt that the individuals that are able to listen and participate here are inspiring the next generation of scientists to really think boldly and bigger. So I will limit my introduction to a single slide that I'll bring up here now, and hopefully everybody can see in presentation format. If anything that I think is important to know about my background is that there's been this thin red line of my career path that has always existed, but my career has meandered to many, many fascinating and interesting paths and adventures. And I'm always excited about what lies over the next hill that might be that next adventure for me. And in doing so, I sort of came up with a couple of clear points that direct me and help me navigate my career and my life in general. I'll honestly tell you, if I knew some of these things much earlier, I'd probably be a lot more relaxed, have less gray hair, and maybe even have some hair in my head left. But if there's nothing else that I can convey to you about what my perspective is and what I've learned, it's these 4 statements here. And that is, everybody should be valued for their perspectives and their approaches. You know, for the past 7 years, I used to work for the Office of Naval Research Global, which is an amazing organization that has existed for 81 years now. And I got to travel the world and look and see what is going on. And my job was to visit academic institutions, companies, incubators, and see what's going on. But the question that I always posed them is that I didn't want to know what they were working on now or what they had published, but what they were motivated and what they really wanted to do. And I learned over time that it doesn't matter if you're the richest country in the world or the biggest country in the world, if you have the passion, the education to do it, and the willingness and the motivation, then your unique perspective and your distinct approach, wherever you might be, is going to be enormously powerful. And that will allow you to do things that nobody else will be able to do. And that's something that needs to be celebrated. I also learned through that and through many other things is that there are no stupid questions. And if you don't ask a question, you'll never know. So you should never be shy about asking questions. I certainly was very shy in the beginning of when I was growing up. Again, if I would have asked more questions, I think I would have maybe been in a different position. The third statement is my mantra. Don't challenge your limits, but limit your challenge your limits. Do not limit your challenges. And I truly believe in that. If we continue to challenge ourselves, we will keep driving forward and creating new things. And alongside that, I think it's extremely important to know that we don't need to wait for a crisis to happen for us to be able to dream up crazy ideas, come up with novel solutions or products or technologies just to solve that problem. Why not do it now? Why wait for that crisis to happen before you can justify doing it? And a lot of times what I learned is that most people think that they need to justify their crazy ideas or seem somewhat reasonable because there's a crisis that they want to try to solve. But that shouldn't be the case, right? We should be developing things as quickly and as radically as possible. And the last one is something that I stole from a friend that I just spoke to this morning. And you always have two choices in life that are not necessarily mutually exclusive, but one takes a little bit more work, I believe, than the other one, and it sort of feeds in with the other, challenging your limits. And that is, if you pursue happiness, happiness is something you have to work on. And it can be uncomfortable, it can be difficult, challenging, with a lot of uncertainty, but it's certainly worth it because it will generate happiness. Versus if you just live comfortably, you might never know what your true potential is and what you're able to accomplish. So these are sort of the statements that I hope you'll take with you, because regardless of who I am or where I came from, those are the things that define me and drive my motivation in the things that I do. So I had the privilege of working for the Office of Naval Research Global for the past 7 years, and you met my successor, Scott Walper, a couple of weeks ago, who presented his perspective. I am now really fortunate to be able to have joined the German Federal Agency for Disruptive Innovation called SPRIND. This is a brand new organization that was created in 2020 by the German government, and it is an organization that invests in crazy ideas, in people who have ideas that nobody else wants to get funded. It's a fantastic group of individuals that is really eager to explore the realm of the art and the art of the possible, but even explore beyond that ledge where we've self-defined constraints, whether in physics, chemistry, or biology, based on the observations that we made. And instead, think bigger and broader than that in terms of what kind of radical innovations can we create? Can we question some of the things that we have established in physics or biology or chemistry as fundamental laws? And can we then create something new or develop new technology based on challenging those conventions? And so this group has been growing over the past couple of years and has been living by that mantra of trying to drive innovation forward. Our founding director is really focused on building a bridge between innovation and entrepreneurship. So you'll hear some really fantastic research today. And I'm sure you yourself will also have opportunities to, at some point, describe the fantastic research that you've developed. The most challenging part is to how do you overcome some of the hurdles and obstacles to translate your idea into something tangible that will affect the world, affect society, and turn it into something, a product that could be something that individuals can consume and use. And so the organization's mission is really to help those individuals accelerate and grow and bloom and become the radical thinkers that they are, and unlock that potential, that genius that is within them. The organization is set up such that we try to fund radical ideas and really try to drive them forward. And we do that in a couple of different ways. So first of all, we definitely invest in the people, because the people are the most important part. You can have an idea pitched by a number of different people, let's say 10 different people, but it will only be one of those individuals that will have the right motivation, that unique perspective, and that distinct approach that nobody else has to be able to realize that idea into something tangible. The other thing that we do is we create the spaces for those individuals to and be empowered to be able to develop the ideas that they want to create, that vision that they have. And we do that by investing in them. What does that mean? Well, what we do is we fund various types of programs. So on the one hand, we do challenge programs where we'll call out an idea that we want an individual to do, or teams to compete against each other for. And we'll do that over 1 to 3 years, where these teams will compete. We'll down select teams, but we'll reward the ones that have been successful with larger volumes of funding to be able to go to the next level with them. And then the other thing that we do is we can also help you create your companies. That is, whether through our challenge or you've approached us independently of the challenge and said, hey, Patrick, I have the coolest, craziest idea that I want you to pitch, and I think it's going to be a breakthrough. What we can do is we can help you fund and develop that company. So what we do is we will actually help you establish that company and invest in a lot of things, including human resources, marketing, human capital, as well as financial, to really help you unlock your potential and the ideas that you have. I'll just go through a quick couple of ideas in terms of our process and how we move forward. We look for those individuals, like I said. I personally believe that 80% of any decisions that I make is about the individual and that perspective that we have. We love taking risks. That is what an organization like SPRINT does. And we're not single-minded in that. There's DARPA, there's ONAR Global, there's the new UK ARIA program, and many other programs. And government plays a very specific role in that, in being able to understand and invest in those bold thinkers that nobody else would invest in. And so we then take the opportunity to support them and encourage them and enable them by providing all of the resources. So we straddle that valley of death where you have great ideas go to if they're not successful. And we then work with the individuals to help understand what they're trying to accomplish, provide them the financial resources, and then help them create the companies that they want. And if we're not taking the risk because nobody else is willing to take that risk, then we would not be investing in those ideas. So the crazier the idea, the loonier the idea, the more blue sky you think that idea is, it's right up our alley because we're willing to take that risk with you. And we're willing to provide the resources if we think we can realize that. And it doesn't necessarily always have to mean that there's a commercial application or a clear commercial application. Sometimes you'll see that we're trying to change the world. So if you're looking at what we're doing at the moment, we've called out a broad spectrum antiviral challenge, as well as a transmission blocking antiviral challenge. There's no market for that at the moment. And we're hoping to create that market by demonstrating that there are successful technologies that can go into many different directions, and that the market then will be really interesting, really interested in supporting. And so that's sort of the perspective that we take in how we try to insert ourselves into the opportunity to make a difference. Cool. So I will stop there with my last slide and just say thank you for the opportunity to present. I'm hoping that as part of this international community, we'll be able to contribute our part with investments, with giving individuals a platform for creating amazing things. And I'll hand it over to Clement now, who is going to talk to you about the amazing research that he's doing.
Fantastic, Patrick. I chose the two Nature journal article that about disruption and disruptive research and technology, and you are a perfect person to basically elaborate what I meant. And I love, what you just said. Also, another thing I want to mention, because I don't want to spend more time. You mentioned last thing about your kind of lesson or advice, and that is interestingly, that is the conversation I did with my friend very recently, and he said or asked me, why you living such a challenging life? And he said he wants to just be comfortable and then easy life. And then my answer was, challenging myself make me so happy. That's my answer, actually. So that reminds me of the conversation I had in his house when I visit him, when I visit the Stanford University for some other thing. So wonderful. Yeah. Thank you so much. That's amazing.
Thank you, Tisa.
Yeah, that's fantastic. Thank you. All right, so let's we'll move on. Okay, so now the main speaker of today with a longer introduction. Dr. Clement Chan completed his PhD training at the Department of Chemistry of MIT, of course, the most challenging institute in the world. After that, he worked with Professor Jim Collins, of course, the synthetic biology father, as a postdoc. In the Collins lab, he published a kill switch paper, which was out a few years before my own work that was recently published. This shows he's really a rising star and creative, and is better than myself. He's currently as an assistant professor at University of North Texas, Department of Biomedical Engineering. His recent work focuses on engineering transcriptional regulators to serve as a genetic sensors and using these engineered parts to explore new genetic circuit design. His research group is supported by several grants from NIH and NSF for this work, including NIH Maximizing Investigators Research Award, one of the most competitive NIH grants. I also believe he is also has a lot of crazy idea. Also, his work is more related to medical application. Also, I'm very happy, now US and worldwide, including SPRINT, thinking about innovation and disruptive technology development, including SPRINT, ARPA-H, and of course the long history DARPA. And I have the pleasure to interact with the ARPA-H people, and I gave lots of feedback to them. And I hope those agencies and also young people nurture new culture that kind of encourage innovation in next several decades or forever. So Clement, thanks for your time today, and I'm so excited to listen to the progress and new direction since the kill switch paper. Please take it away. Thank you.
Okay, thank you. Thank you very much for the nice introduction and this opportunity to share some of my recent work. And also, thank you, Patrick, for this very inspiring talk. So challenge your limit and not limit your challenge. I think I'll put it on the wall of my lab and show it to all my students. Yeah. All right. Okay. So let me share my slides. Let's see. Sorry. Right. All right. So, oops. Sorry. I guess there's a freeze. Sorry. Let me just start it over again. All right. I hope everyone is seeing my first slides.
Yes.
Okay, great. Yeah, so today I'm going to talk about some of the recent work that I've been doing the last several years to engineer some transcriptional regulators and how we can use them to engineer cellular behaviors so that these cells can be used for different applications. So transcriptional regulators is one of the oldest topics in synthetic biology, I would say that. So people start to actually discover these regulators since 1960s. One of the earliest one is actually discovered by Jacob and Mono, which is the LAC-I system, and they actually get a Nobel Prize out of this. So for this regulator LAC-I, what it does is it actually binds to a DNA promoter to repress gene expression. But then with the presence of the small molecule inducer IPTG, then the protein detached from the promoter, which then allow gene expressions to occur. And still up to date, these regulators are some of the most commonly used genetic sensors for building genetic circuits for controlling cellular behaviors. And after the development of the LAC-I system, scientists have discovered many of these regulators with similar structures. So they call these all regulators together as the LAC-I superfamily. For all these proteins, they have a DNA binding module which bind to a specific sequence of DNA. And they also have a regulatory module which binds to specific small molecules. And the binding of these small molecules effectors can then affect the binding affinity of the protein to the DNA. And so with these behaviors, then these proteins can be used as sensors. So how it actually works is also very straightforward. So these proteins, it binds to a part of the genome, which is the promoter, which then it blocks the transcription to occur. But then with the presence of the small molecules inducer, these small molecules would then trigger a change in the conformation of the protein, which then these proteins detach from the DNA, which allows the promoter now be interacting with other proteins for transcription. So you can see that the system is very simple, and most of us have used some of these. And just with one protein, you can then actually letting the biological system to detect one specific signaling molecules, and based on that to control the expressions of a specific gene. And so actually we know there are many transcriptional regulators, and then they can sense a wide range of different small molecules, from something as simple as a single metal ions like sodium ions and zinc ions, to some more complex molecules such as fatty acids and other bigger macromolecules. So potentially we can use these regulators to sense many different biological components or many different biological signals. But then one problem that we always face in using these transcription regulators is that they are actually not modular. They are very rigid. So what I mean rigid and not modular here, I can use this to explain to you. So here, for example, if we have one regulator, we can use it to control the expressions of a gene, such as like control the promoter A by using a small molecule A to do that. So this is one connection between the small molecule or the signal and a specific promoter. And for example, if we have another regulator, this is, let's call the regulator B, it can then actually form a connections between the signal B to a promoter B. So this is great, in the biological systems, we want them to be specific and they are not actually interacting with each other, no crosstalk. But then if you think about in the engineering perspectives, a lot of times we want them to be more flexible. So here, for example, if we want to use a small molecule A to control gene expression driven by promoter B, this is currently not possible in many cases. And so this is what we call it to be not flexible. And it's actually become not modular. And because of that, it actually limits a lot of genetic circuit topology that we can actually implement or realize. And so when I start my postdoc at James Collins Group, I have this idea. So I'm thinking if we can build some of these hybrid regulators, then potentially we can use them to build new connections between different signaling molecules and different promoters. So here, for example, if we can fuse a ligand binding module from this regulator A with the DNA binding module from this regulator B, then potentially we can generate a new hybrid regulators that can actually sense the small molecule A as an inducer, and then it can bind to promoter B to control gene expression. And then with these hybrid regulators, we can then now build a new connections between the small molecule A and promoter B. Okay, so this is the idea. And then by studying the structure of these proteins and then looking at their biochemical properties as well as their biophysical properties, then we actually come up with like a part of the proteins that we think is a good DNA binding module, and another part of the protein, a conserved part of the protein, which is a good small molecule sensing module. So one of the earliest experiments we have done is here we actually combine LAC-I DNA binding module with the small molecule sensing module from a protein called GALR, which sense galactose as an inducer. And here you can see that with this hybrid regulator, when we express it in some E. coli cells that contains a GFP gene that is driven by the LAC promoter, you can see that cells contain the system, when they're exposed to galactose, there's a dose-dependent increase in the expressions of the GFP, which shows that with this new system, it can control gene expression driven by the LAC promoter, which is a property from this LAC-I protein. And then it can detect galactose as an inducer, which is a property from GALR. And so it shows that our idea is actually working. And so after I start my own lab, I continue to explore the development of these hybrid regulators. And here is a set of 40 hybrid regulators that we have built in the last couple of years. So here in this set of hybrid regulators, we are using 5 different ligand binding modules that can sense 5 different signaling molecules. And we fuse them to 8 different DNA binding modules, which generate this set of 40 hybrid regulators. And here with these 40 hybrid regulators, you can see that about half of them are working pretty good. So actually, in fact, 22 of these hybrid regulators out of the 40 can generate fold induction of the GFP of above 10 folds. But then the rest of the half, which includes 9 regulators that only generate less than 10-fold of induction, and also another 9 that actually generate less than 3-fold of induction. So taking this as an example here, if we use the GALR DNA binding module and fuse it with the LAC-I DNA binding module, even when the cells are exposed to IPG, you almost see no induction. So these are some of the proteins that are not working. But with some of them are working very well. So you can see, for example, this one we use a LAC-I DNA binding module with a cell-out small molecule sensing module for sensing cell lobules. You can generate over 200-fold of induction. And also some of these like down here. Okay, so here it comes with the new question. So we see that half of them are working, but the other half are not working so well. And as a scientist and an engineer, we think about why some of them are working so well, but some are not working that well. So this is actually the main question that I will try to answer and show it to you in the very last part of this talk. But before I actually go to that part to talk about like how we further engineer these proteins and understand why some work, some does not work, I would like to show you two genetic circuits that we have built by using these hybrid regulators. So a lot of people, like when they see my hybrid regulators, they will say that, oh, very cool, you know, like looks like you can like have some new regulators with new functions. But why would we need these regulators? This is actually usually the first questions I got. And here, like with these two genetic circuit and also the circuit design, hopefully like it would show you like some interesting and new properties that we can harness from these hybrid regulators for developing new cellular response functions. So the first device that I would like to show you, I call it multiple toggle switches with a master off signal. So with this toggle switch, I guess for many of us, we know what is a toggle switch. So it's one of the classic genetic circuits that people have been studying for many years. So with a genetic toggle switch, what it happens is it requires to have two transcriptional regulators, or I should say transcriptional repressors. So the two repressors, they repress expressions of each other. And with that, you can generate two stable states. And with the two stable states, you can use it actually as a memory device. So what I have done here is I'm building actually multiple toggle switches within the same biological system or within the same cells. So for example, here you can see that I have two toggle switches here. So for these two toggle switches, each of them can sense different signal molecules for turning on the expressions of different reporting genes. So for the one on the top here, it's actually detect IPTG, and with the presence of IPTG, it would then generate the M cherry, which makes the cells to become red. And for the other toggle switch, it's detect ATC and hydrotetracycline, which is also a very commonly used inducer. And with the presence of these signal molecules, the cells will start continually making GFP and turns the cells to become green. And so what is difference for this toggle switch versus the previous toggle switch is you can see that for these two toggle switches, the counterparts of the repressor is actually, one is the MOR RBSR, which is a regulator that can detect ribose, and then it can control gene expression driven by this promoter MOR-O. Versus for the other transcriptional regulators, a native RBSR, it's also detect ribose, but then it's control gene expression driven by the RBSO promoter. So you can see that for these two regulators, they detect the same small molecules as an inducer, but then they actually control a different promoter. So there is actually no crosstalk between these two regulators in terms of interacting with the promoter or the genetic system, but then they share the common signaling molecules. And so with these systems, what you can expect is when you expose IPTG, the cells would turn red, and you expose the cells to ATC, the cells would turn green. But then you can use ribose to actually reset both of these toggle switch back to the stage that they don't express the reporter anymore. And here are our results that we published several years ago. So here you can see that indeed when the cells are exposed to IPTG, the cells become red. And when the cells are exposed to ATC, they become green. And when cells are exposed to both IPTG and ATC, they become both red and green. And so once they turn to that color, then after we remove the inducer, they can remember that exposures and continuing generating the reporter gene. But then in all these stage, when the cells are exposed to ribose, they all turns back to become not red and not green. So it shows that we can use the ribose as a master signal to control both toggle switches. So it shows that the idea is working and this is a new behavior. But then again, why would we want to have this kind of behavior? So some other scientists propose to use these toggle switches to actually sense multiple physiological signals in a biological system. So for example, on this picture that I actually get from these review articles from my postdoctoral lab. So it actually shows that, for example, we can have a bacteria for sensing 4 different physical parameters or biophysical parameters within in the inside like a biological system. And then based on actually which toggle switch is being turned on for generating the reporter, you can then know what is happening within the biological system or within the host. But then with all the conventional toggle switches, you can imagine that if we have 4 different toggle switches for sensing 4 different signals, then with this cells, if we want to actually reset them, you would need to use 4 different signaling molecules to reset all the 4 toggle switches. But then with what we have developed, if we can have this hybrid transcriptional regulators, and then for so that like all these toggle switches can share the same common resetting signals, then you only need to have 1 signaling molecules for resetting multiple toggle switches, which we believe is an advantage. It makes the system easier to control and also more reliable. All right, so this is the first genetic circuit that I want to share with you. And the second genetic circuit system is actually what Tae-Shuk has mentioned, which is the biocontainment system that I developed during my postdoc time. So we call it a programmable passcode circuit. So for a biocontainment system, the idea is that when we develop some engineered organisms, we don't want them to be proliferate or spread to the natural environment. And of course, we don't want them to actually get into any human or any animals. So because of that, a lot of scientists has tried to develop this biocontainment system, which then constrain the engineered organisms to only survive under very specific conditions. And when they leave the condition, for example, if they go into the soil, the river, or even get into the stomach of a kid, then because the environment has changed, and then these engineered organisms would die. So this is the idea. And for our biocontainment system that we developed with these hybrid regulators, it can detect 3 different signaling molecules. So with 3 different signaling molecules, with either the presence or absence, we can have 8 different combinations. And cells containing this biological containment system can only survive under 1 very specific combinations of the 3 inputs. And we call this the passcode for survival. And how we can actually get this behavior is with these genetic circuits that I showed here. So in this circuit, you can see that we have 2 hybrid regulators. Both of them can bind to the same promoter, but then detecting different signaling molecules. And then for these 2 regulators, they actually repress the expressions of a 3rd regulator. And this regulator would actually bind to this purple promoter to repress the expressions of the toxic gene. So with such a system, you can see that for cells to survive, which means they cannot have the expressions of the toxic gene, then it would require the presence of both this green signal and this yellow signal, so that this 3rd transcriptional regulators can be expressing, which then repress the expression of the toxic gene. And also the cells cannot expose to this blue signaling molecules, because these blue signaling molecules would then actually induce the expressions of the toxic gene, so the cells would also die. So you can see that with this circuit, the cells would only survive and not expressing the toxic gene with the presence of this green and yellow signaling molecules and the absence of these blue signaling molecules. And what I think is very important for this circuit or this design is it is very easy to reprogram the signal for survival or the passcode for survival. So on top here, this is one of the genetic circuits, which is the one that I showed you on the last slide. So it requires to have green and yellow to survive, and also the absence of blue. But then if we change the signal sensing modules of these 3 regulators, then you can easily change the passcode for survival. So here, for example, if we use these 2 regulators to control the expressions of these green regulators, then the cells would require the presence of yellow signals and blue signals and the absence of the green signals to survive. And this is important because for these genetic circuits, we expect to be used in different applications, which the engineered cells may need to perform their functions under different conditions. So if, for example, like in one conditions that the cells definitely requires to have the blue signal to be present, then obviously we cannot use that for the passcode for survival. Then we need to change it to some other combinations of the signals. But in this system, you can see that we can build all of them quite easily and changing the survival conditions. Okay, so these are the two genetic circuits that I would like to share with you. And currently, we are actually trying to use these hybrid regulators to building more like circuit topologies. But with the limited amount of time that we have here, I'm not going to go over those. That is more preliminary data. And so now let me go back to actually the question that I proposed earlier. So as we see for these hybrid regulators, some of them, they're working very well, but some are not working so well. And we want to understand why this is the case. So when actually I show this data to many of my colleagues, they will say that, oh, 50% of them are working, which is good enough, like potentially like we can use those 50% to build many circuits already. But then for me, I'm thinking actually this probably may not be sufficient. Just thinking about like if you have a keyboard, so most of the keys are working, but then you just cannot type the letter A, then this is actually very inconvenient, right? And same here, if we can use like a lot, build many connections, but then still there are some connections we cannot build, then it's still potentially limiting a lot of potentials for us to develop this genetic circuits. And also, as a protein scientist, I'm just very curious. So I would like to know why actually some of these regulators are working so well, but others simply does not work. And with this interest, then I actually spent the last 2 years trying to understand why some of these are working, but some of them are not working. So this is our hypothesis. We believe that for these hybrid regulators, for them to work, they must maintain some critical interactions between the DNA binding module and the ligand binding modules. So for these interactions, over the evolution of these regulators, some of them may need to be changed. So for example, this I1 may need to be changed so that the new regulators can bind to a new sequence of DNA. But then if this I1 is being changed, then it may lose some very important interactions with the ligand binding module. But that would be okay because those residues on the ligand binding modules that are interacting with I1 can co-evolve. So they will be changing simultaneously. So for the purpose of maintaining these important interactions. So that's why like actually in the nature, just talking about the LAC-I regulator family, we have over 70,000 of them. And we assume like since we find them in the nature, all of them are working. But then now when we develop these hybrid regulators, we basically don't let them to evolve, but we just basically cut away, for example, the ligand binding module and swap it with another ligand binding module from another protein. And then in that case, a lot of these interactions potentially can be lost. And if they are lost, then obviously the protein does not work. So looking back at those 40 regulators that we developed, half of them are working, which we can say that because most of the important interactions are still maintained within them, but the other half does not work, probably because some of the important interactions are being lost already. Okay, so this is our hypothesis, and then based on this, we are developing this computational model to test our hypothesis. So what we have done is we first align over the 70,000 LAC-I family members, and after that, we then fit them into actually a program to trying to looking at the co-changing of those residues between the DNA binding module and the ligand binding module. So if two residues, they always co-change, then we just assume that they are actually, they maintain a very important interactions between the DNA binding module and the ligand binding module. And then after we actually collect all these interactions, we then put them into this mathematical model and use it to compute a score. So if all these important interactions, they are maintained, that hybrid regulators will get a good score. But if like some of these interactions are being lost, the score would decrease. And then here we're trying to look at if the scores is actually complementing or supporting the experimental data that we have observed. And so here are the results. So for the experimental data, I've already shown you several times. And on the bottom of this picture here, here is actually the computational prediction results. So here, when we compare the experimental results with the computational results, we see that 35 out of the 40 regulators are predicted correctly. Only these 5 white ones are predicted wrong, which means, like, for example, in this case, we think this may be a good candidate, but in fact, this is not working. So this is what we think is predicted wrong. So there's only 5 of them are wrong. So this actually gives us an accuracy of over 88%. So with a computational predictive model, we think this is actually pretty good, and it supports our hypothesis of why some of the proteins are working, but some are not working. Okay, so then now it comes to actually the exciting part, or at least I think it's exciting. So we try to use this model to rescue some of these hyperregulators. Okay, so at this point, what we see is if the protein get a low score, then it is basically not working. But if it's get a high score, then it is working. Then potentially what we need to do is we just need to change the sequence of those hyperregulators and then fit in the model again until it's get a good score. And this is exactly what we have done. So what we have done is we take one of these hyperregulator that is not working so well, which is the LECI RBSR. And then we just generate all the possible single mutations for this protein. So for this protein, there are about 300 amino acids, and each amino acid has about, has exactly 20 different possibilities, because there are 20 amino acids. So 300 times 20, it is only about 60,000, which for a computational program, it is actually not bad at all. So we just submit all the 60,000 sequences to our predictive model and then see which one would get us a better score. For those that give us some of the best scores, we combine them to make double mutation and also make triple mutations, so that we can get proteins with much improved scores to show that, like, the ligand binding module and the DNA binding modules are actually compatible. And with all these hyperregulators mutants that we developed, then we experimentally test some of those that have the best scores. And here are some of our results. So this is the LECI RBSR that I just described. You can see that originally with the hyperregulators, it only generates about 6-fold of induction, which is not very good for building genetic circuits. But then with those mutations that we predicted, and here is only one single mutation, a K57A, the protein become much better, and the performance of gene expression control is much better. So you can generate over 200-fold of induction. And also the background expression, the leakiness of the system has also been much reduced. And it's not only for this LECI RBSR, for several other hyperregulators that we have been using this approach to engineer, they also works. So for example, this PRGRL, you also see great improvement, as well as the other two at the bottom. But here I would like to tell you that it's actually not always working. So if you check out our paper published in 2021, you'll see that these are actually the only 4 that are being improved with this method. In this paper, we actually describe, we're trying to use this method to engineer 8 hyperregulators. 4 of them can get improvement, but the other 4 cannot get it. But due to the time limit, I'm actually not going to discuss too much about that. But I just want to show you some other results for why actually some of them is not working. Here you can see that this is another system that we try to engineer, which is the TET-R transcriptional regulator system. And in this system, what we're trying to do, again, is build hyperregulators. We are trying to use the TET-R DNA binding module and fuse it with the MPHR small molecule sensing module. With these hyperregulators, you can see that, again, it has some biological activities, but then, like, these activities is not as good as what we want. At least it does not get to maybe 100-fold of induction. And then we try to use the similar idea to try to engineer the regulators and improve its performance. And here, if you look at the molecular level, for the TET-R, we found that one interactions between the ligand binding module and the DNA binding module is actually involved like this carbon chain, side chain. so which believe it is mostly hydrophobic interactions. But when we look at those interactions for MPHR, you can see that on the ligand binding module side, this is a threonine, which has a hydroxyl group. And then on the ligand binding module side, it has an arginine, which is partially charged. So the interactions is more likely to be not hydrophobic. So it's more polar interactions. So because of that, you can imagine if we fuse the DNA binding module from TET-R with the MPHR ligand binding module, then you are forcing like a hydrophobic carbon chain to interact with some partially charged amino acid residues, which is not efficient. And so we have tried to engineer it by changing these residues. And here is like some of the mutants that we have developed. Among this mutant, we see something quite interesting. So here, for example, for this mutation that we change the arginine to an alanine, you can see that the binding of the protein to the DNA has been improved. So the leakiness has decreased. But then actually the proteins would lose its functions for sensing the small molecules. So in that case, like we can say that it has actually have improvement in terms of recognizing the DNA sequence, but then it's actually lost its functions of sensing the small molecules. And based on these results, we then come up with updated model or hypothesis. So we believe that just now we say that like there's some important interactions between the DNA binding modules and the ligand binding modules. So yes, I think this is correct based on what we see. But then now we also think that for some of these residues that are involved in the module-module interactions, they may have another functions. So they may also be involved or critically involved in interacting with some other residues within its module. So here, for example, like J2 may be involved in interacting with this J5, which is on another part of the ligand binding module. And so in this case, if because, for example, I1 has been changed, now I need to change J2s to actually to rescue the interactions between I1 and J2. But then changing J2 may actually cause the loss of the interactions between J2 and J5. And so this is why like the proteins sometimes like even after the modification or after the mutation, they still does not work. And so because of that, we have used similar strategies to develop another computational model to give us a score for predicting the impact of a mutation on a module for interacting with other residues within the same module. And we call this the SF or a structure-based score or SFS score. And here are actually on the right-hand side of this slide here are the results that we have got for all these 8 proteins. So the top 4 ones are the ones that I've shown you that we can rescue them, and the bottom 4s are the ones that we cannot rescue them. And here you can see that for those that we can rescue, which are actually for those mutants, you can see that they are in red. They are the ones that actually we see like an improvement in terms of the regulator activities. So for all these 4, you can see some of them has red dots, but then in the bottom one, we don't see any red dots. And what you can see that for all those that have these red dots, which means like they have been improved, these mutants, if you look at the SFS score, they are actually having a more favorable score compared to the original ones. So this may be a little bit of above this line, which means the score is slightly worse, but compared to the others, they are still better. So what it's actually telling us is now with the SFS score, we can further predict that for these mutants, after we mutate them, whether they would be able to maintain its original protein structure or maintain the original protein stability. And you can see that for the bottom 4 ones that we cannot rescue by actually using the CS score for the mutation, which is the first predictive model, we cannot rescue them. You can see that all these mutants, their SFS scores is less favorable than the original hyperregulators, which then is explained why like we cannot rescue them. Okay, so with all these results that I'm showing you, here is a streamline of how we now actually engineer or design and generate these genetic sensors. So first, we based on the structural similarities, we can then first have like the first sequence, which is like how we develop those 40 hybrid regulators. And then with these sequences, we can then first actually use the CS score model to look at the compatibility between the ligand binding module and the DNA binding module. If it gets a good score, then we can move on. If the score is bad, then what we need to do is then we have to generate mutants and then fit them in and see if you can improve it. And for those that have a good CS scores, then we can now use the SFS score model to see if those mutations is going to affect the structural stability. And if they also pass that, then we can then test them experimentally. And so with this streamline, we are currently actually exploring the generation of these hybrid regulators in many transcriptional regulators. So most of the results I show you today is from the LACI family. I briefly show you some results from the TET-R family, but also now we are actually expanding it to other regulator families. So this is what I would like to share with you. And at the end, I would like to thank those people who have been contributing to this project. So as I mentioned, this project or the idea first started when I was a postdoc in James Collins Group. So definitely have to thank Jim for supporting that. You know, like he obviously like I'm very glad that to be joined his group. His group is just kind of like a theme park for me. There are so many toys to play with. And also John, Ewan, and Caleb are involved in this project initially. And after I start my own group, then I now have very great collaborators. So Farouk is a computational biologist who actually do a lot of this computational program development with me. And Shani was his grad student, but he's now a postdoc. And also I'd like to thank my collaborator at UT Tyler, including Joe and Dustin. And here, these are my current lab members. I think I have a very good group. I'm very fortunate. And you can see like these are the people. And I would like to thank Ray. Ray is actually a lab technician in my group when I first started my group. So without him, probably I would be still struggling to publish my first 2 papers. So I specifically put his picture here. I think he definitely is a very important person in my lab. And also like there are some previous members also involved in this project. And also at the very end, I would like to thanks for all the support for this group from these funding agencies, including NIH and NSF. And specifically, I would like to thank Mike and Dave, who are my program officers. And they give me a lot of great advice to improve this project so that it can be keep moving forward. And I'm also very thankful to Taeshok to giving me these opportunities. I think I said that again, but I want to say it again. Very glad that I can be sharing this with like all these people in different parts of the world. And very happy to answer any questions. Thank you very much.
Fantastic, fantastic. You know, advance a lot. And one thing I love is, 50% people may say, I mean, that's good enough, but you challenge yourself, as Patrick already said, and then you basically come up with the new idea to solve the complex problem using something else. Fantastic. Patrick.
Yeah, I really love the term challenge your limits. Don't limit your challenge. Yeah.
Yes, I love it.
I probably will keep telling my students for the next couple of days.
Okay, that's good. So Patrick, do you have any comments, question?
No, it's amazing work. I really like. I think it's a perfect example of how you're applying your unique approach and distinct perspective on a very challenging topic and applying what you think is the best way to do it. So it's very cool. Congratulations.
Okay, thank you.
Fantastic. Let me check whether we have any question from audience. Okay, I have one person raise your hand. So let me allow Javier to talk.
Hello, can you hear me?
Yes, please go ahead.
So nice talk. So I have a question regarding the engineering of the TET-R regulator family. So because I think I saw in the paper that actually when you change the ligand binding domain, you also need to change the spacing that is between the palindromic sequences in the operator. So I guess this in a way compromises a bit the modularity, right? Because when you change the ligand binding domain, you also need to change the operator. So I was wondering whether there's a way of bypassing this, and also whether this also occurs for the LACI family or not.
Yeah, I think this is a good question that I did not mention about that. So let me just go back to that slide. So I think Jaffer is mentioning about this system here. So actually, we have published two papers in 2019. One is on the LACI family, and the other one is on the TET-R family. For the LACI family, it's just because of the flexibility of the DNA binding module. We do not need to change the spacing for the binding. So at the spacing, I guess I need to explain a little bit. So you can see for this structure here, for example, this purple part is binding to this major groove, and the other purple part is binding to the other major groove. And actually, this is a dimer, a homodimer, which means that you have two peptides are exactly the same. They form one protein so that they have two of the exact same group, but you can see they bind to two different sites, two different sites within the DNA. And then you can see there's actually kind of a spacer within the DNA between the two binding sites. So here is the spacer. So with the TET-R family, it is true, like in that paper, we see that we actually need to actually change the distance of the spacer for different protein. So for example, the native TET-R versus this TET-R MPHR, the spacer would be different. And the reasons for that is because for this protein, the TET-R family, you can see that it is all with alpha helix, which actually this structure is very rigid. And then this ligand binding module is actually dominating the distance between the two, this we call the helix turn helix motif for binding the DNA. And so we need to change it. So I think Jaffer's question is for this system, is there any possibility we can actually engineer the protein so to make them to be, the spacer can be constant, so we don't need to always change it? I think that is the question. And yes, thank you. And so for what we see right now is seems like we cannot do that, because if you need to change the spacer to make this to be unique, you basically need to have like sophisticated engineering of the whole structure of the ligand binding module, so that the ligand binding module can be constantly within that range. We cannot do that, but I think this is actually something I keep asking myself and see how we can deal with it. Right now, we know that we still cannot go to that phase, but what we are now studying is we're trying to actually study this ligand binding module, and based on the structure that is already published, and also based on like what we can predict the structure, we try to use that to predict which of this ligand binding module will share the same spacer. So instead of we engineer it, but we just predict what would be the spacer. So for example, we can predict that for MPHR, the spacer may be 2 nucleotides, and then for TET-R is only 1 nucleotide. So we can predict that. And then after the prediction, we can base on that to see if we can develop a set of these hyperregulators that can use promoter with the same spacer.
Okay. Go ahead.
Yeah, thanks for this question.
So Javier, go ahead. Okay, so we have another question from Maya. The residue between the LBM and DBM, do you have any sense of whether it's stabilizing the LBM and DBM, or if it's involved in some way in the signal transduction that cause binding? And she also adds, basically, if you stabilize LBM and DBM separately, would that increase modularity or destroy the cause and effect?
Yeah, I think this is a very good question. So even with one protein, we know that when we go to this molecular level, it's just becomes so complex. So we're talking about the interactions between the two modules, and now we talk about the interactions within each modules for stabilizing it. I think she's thinking in the right way. So it is actually important to think about like each of them. So I think I take this as a for a system like that, I take it as if when we get the ligand binding module or DNA binding module by itself, it is actually stable enough that it should maintain its structure. The reason why we actually need to now go in and change it or modify it is because when we actually try to modify the interactions between the two modules, you actually disrupt some of the interactions within each of the modules. That's why we need to go in and change it. So we did not try to just go and say, oh, so we have a protein that is already working, and then we just go in and try to change the stability to see if it would be better or not. So I have one assumption, and actually, if you look at the coevolution model, the whole assumption is that all these proteins we find in the nature, they would already be in their optimized form. And so we only needed to change it if we need to change it. So we haven't actually go into the stage to just change it and see if that would be improving the stability. I hope that answered the question. Okay.
Okay, we have another question from you. How's the specificity of LBM when different domains are combined? Are they more promiscuous or still very specific with the original ligand?
Yeah. Yeah, I guess this is a question when we submit the paper is obviously the question that the reviewers would also ask, because in synthetic biology, like the interoperability and no crosstalk is a very important aspect. And at least for what we have shown, like for example, in those regulators, the 40 regulators that we developed, we have tested whether there's crosstalk or they would actually have any shared response to the different inducer. And we see that both the interactions between the DNA and the protein and the interactions between the protein and the small molecules are very specific. So after changing it, it actually does not cause any change in these properties, which I think it makes sense, because if you think about that, for example, like the question is like if we change the ligand binding module with different DNA, would it actually be able to sense other small molecules, which the chance actually is very small, because you have your whole binding pocket within the ligand binding module to be the same. So even if it is changed, it would not be easily like fitting in molecules that you can be found in the limited small molecules that is exposed to.
Okay, let me check whether we have any question, because now I see 11, 12. I have a question, but I will just wait for our informal chat. So let me close. Okay, so thank all for joining and staying today. We'll meet again on March 30th, Thursday, the same time, same Zoom link. We'll have Professor Victor Del Lorenzo at National Center of Biotechnology, Madrid, Spain, and Professor Junyoung Park at UCLA. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with today's speakers and me, and I will promote you to panelists who can speak and show your handsome and pretty face if you wish. Thanks, and stop recording.
Thank you, everyone. Thank you so much.
Thank you. Stop recording.